Hello, and I'm sure that you all are well versed with the generalized structure of virus. What are its various characteristics? How does it differ from the bacteria, fungi, or a human cell? How does it replicate, and what are the basis of the classification of the viruses into various groups? Now, we all know that the viruses, they are obligatory intercellular particles. Uh, they contain either the DNA or the RNA, and all viruses, they have the host cell tropism, that they have the specific affinity for the specific cell in the host. They cannot replicate at their own because they lack the metabolic machinery that is required for the synthesis of the macromolecules and the production of ATPs, the energy which is required for all the functions. That's why the viruses has to depend on the host cell machinery. It enters in the body, it takes over the host cells, and it started structuring the host cells to make the proteins of the virus only test. It structures protein, its functional proteins. And we classify the viruses based on to the various, its symmetry, based on to the composition of the genetic material, whether it's a RNA or it's a DNA. The virus, it will never contain both the DNA and the RNA. It contains either the DNA or the RNA, and this unique composition of the viral genome has been exploited by the scientists for the classification of the virus. So we have the DNA viruses and we have the RNA viruses. Today we will study a virus which is a RNA virus, and this virus belongs to the family what we call as the rhabdoviruses. The rhabdoviridae are the rhabdoviruses. These viruses, the rhabdoviruses, they are single standard negative polarity viruses, RNA viruses. Now the virus, we have the double standard RNA viruses, we have the single standard RNA viruses. The rhabdoviruses is a group of viruses which are single standard negative polarity RNA. Now, the negative polarity RNA, before it's being, the transcribed, or it has to enter into the replication, it has to be changed into the positive polarity RNA. So all the negative standard RNA viruses, that has to be changed itself before it's translation to the positive standard RNA virus. They have to carry their own RNA-independent RNA polymerase. Now, this virus is a bullet-shaped virus, and perhaps one of the biggest viruses. Pox virus is the biggest one. This you see the virus, and it's surrounded by a lipoprotein envelope. So this is not a non-enveloped virus. It's enveloped viruses. Rhabdoviruses group are single standard negative polarity RNA viruses, which are bullet-shaped, and they are enveloped viruses. The viruses of this group, they have just single antigenic type. This means there are no sub-variants of these viruses. There is one virus that provides, one type of antigenic position, and this antigenicity is in the glycoprotein spikes. This glycoprotein, if you look at this, the outermost layer. The outermost, this is what we call as the glycoprotein layer. This is the matrix. And this is you see, this is how the nucleoproteins, ribonucleic acid, RNA viruses, shaped like a bullet. So this has just a single or one antigenic type, and this antigenicity is based on to the glycoprotein composition of the virus, and it has a broad host and can infect all the mammals, including humans, animals like buffaloes, and other animals also. So broad range of host, single antigenic composition, single standard negative polarity RNA viruses, which is enclosed within a bullet-shaped capsid. Capsid, there is a protein coating of the virus. And this is what we call it, the capsid. The virus is nothing. You see, it's just a particle of DNA or RNA which has been enclosed in the protein. So if you have a protein and you have the genetic material in the form of DNA or RNA, you can make a virus, but it's not that simple. It really requires something. But we can make it if you can try it. So the structure of the rabies virus, the rabies is one of the important member of the rhabdoviruses family. This is how it looks like, that there is a glycoprotein There is a glycoprotein, which are the outer spikes. You see, these are the head. This is spike. This anchored into the matrix, and we have the nucleoprotein inside. So this, and look, the bullet-like structure. So glycoprotein spikes, and these spikes, I'm sure you must have studied that how the virus, they attach themselves to the specific receptor sites in the body. The specificity of viral tropism is very important in the pathogenesis of the virus. If the virus does not attaches itself to the receptors present on the specific sites in the body, it will not be able to replicate, and it will be just wasting its life. So this is very important. In all viral diseases, the glycoprotein spikes, they are very important because they provide basis for the adherence and attachment of the virus to the specific tissues in the body. The replicative cycle of the rabies virus is very interesting. You see, the virus, after the bite, it enters into the human cell, and it attaches the acetylcholine receptors which are present on the cell surface. Now, what happens then after attaching over there? The variant RNA polymerase is synthesized the 5 mRNAs. Now, these 5 mRNAs, they code for the specific viral proteins. Now, virus need two types of proteins for its replication. Number one, it needs the structural proteins and it needs the functional proteins. So the viral genome is not that much long. It's just 5 genes over there, and these 5 RNA genes, some of them will make the proteins, and some of them will make the functions of these proteins available for the viruses. So the virus cannot replicate at its own. It has to enter into the host cell machinery, take over the charge, and instruct the host cell machinery to make the proteins that are intended for its virus. After replication of the genome, the progeny RNA viruses, they assemble the proteins and to form the nucleocapsid. And then, of course, in order to protect this nucleocapsid, we need the envelopment is required, and the variant bites through the cell membrane. So when our virus, they leave the cell, they take some of these cell membrane with it, and that cell membrane make the envelope of the virus. Transmission and epidemiology of the rabies virus, we all are well versed that it is mostly the bite of rabid animal that has been infected by the virus, and the infectiousness of the rabies virus in dog is being manifested by the aggressive behavior of the animal. It's biting behavior induced by the virus encephalitis, because the virus has entered into the dog, reaches to the brain, causes encephalitis that makes the virus, that makes the dog mad. And when bite, it just leaves the virus inside. This is what we call the so-called non-bite exposure aerosol of the bat has also been reported. So the dog is one of the reservoir and the transmitter, but that requires the bite, of course. But the non-bite exposure aerosol from the bat has also been reported, and it does contain the rabies virus. Another real example of the transplant of cornea taken from a patient who died of undiagnosed rabies. This means that the rabies virus can also be transmitted through the transplantation of cornea. You have to keep in mind. The hundreds of cases in developing countries like in Pakistan, we have many, many cases. We have a huge population of stray dogs, and you never know who one is infected. And that's why we have many cases over here. The United States and the other developed countries, they almost have been declared canine rabies free back from 2017. Approximately worldwide, there are still 50,000 people that die because of the virus. The rabies virus is cell-causative agent for the death of almost 50,000 people worldwide. So still a very interesting and a very important clinical issue that we have to address, particularly in our setup. In Pakistan, we don't have much protection from the stray dogs, and incidents are increasing, and we have many, many incidents of rabies bite every year, every month, every day in Pakistan. Now, if you look at this diagram, it will just to let you know the sketch of why the virus, how the virus enters, and how does it spread in the human body. This is how you see the virus, it enters via the bite of the animals. And then the virus, it replicates in the muscle at the site of bite. So virus enters, it adheres, it is absorbed over the cell, and then it starts its replication in the muscles. Now, virus then, you see, it then infect the nerves, particularly the peripheral nervous system. And it moves by the retrograde fashion from the axon to the central nervous system. And the virus replicates in the dorsal root ganglions. So the replication, the site of predilections, or the site for tropism for virus is the nervous system, particularly the dorsal root ganglion. And then it travels to the spinal cord and then to the brain, where you see when the brain is affected, there is meningitis. And then you see there's virus travels from brain via nerves to all the tissues, go to eye, can go to kidney, can go to salivary glands. So this is how the generalized picture of virus sentient spread in the human body look like. Bite by the dog, virus replications, then traveling through the retrograde fashion, goes to the brain, causing meningitis, and then from meningitis to the other tissues of the body like kidneys, salivary glands, and eyes. Now, this is how the pathogenicity and immunity would develop in case of rabies virus. The virus, after entrance of the bite, it multiplies locally at the site of bite. And then you see after multiplication, it infects the sensory neurons and moves to the central nervous system by the help of axonal transport. The virus is sheltered from the immune system with little or no immune response because the virus never enters into the bloodstreams. So the until or unless an infectious agent, maybe virus, maybe bacteria, maybe fungi, they entered into the bloodstreams, or they elicit the immune response will never be there. Because the virus is sheathed and it enters and travels through the nervous system, so there is no immune response. So we cannot detect the antibodies in the blood. So then it multiplies in the central nervous system, travels down the peripheral nervous system to the salivary glands and the other organs also. Enter the saliva to be transmitted by the bite, and there is no viremic stage because the virus does not enter in the blood. There is no viremia. There are no antibodies. There is no cell-mediated immune response. The virus protects itself in the immune system sheath. So keep in mind all these things. Now within the central nervous system, it will cause encephalitis with the death of the neurons and demyelination of the nerve cells. Infected neurons, they contain an eosinophilic cytoplasmic nucleons, what we call as the Negri bodies. Negri bodies in the brain tissues. In the brain tissues, they are pathogonomononucleons for the rabies. You make the slide and you see the pathogonomononucleons are present over there. But some of the hosts, they may not show these pathogonomononucleons like Negri bodies. This is important about a diagnostic for the rabies. We have to rely on to the detection of antibody and the Negri bodies. It's not an antibodies. It's a nucleon. It's a cytoplasmic nucleon of the cell, which occurs in the form of eosinophilic cytoplasmic nucleons, which is called the Negri bodies. So pathognomonic brain lesions of rabies, they are known as Negri bodies. Now, this is how you can see the rabies histopathology, which is very important. The perivascular cuffing or the inflammation around the blood vessels. There is perivascular inflammatory cells over there, cell infiltrate, and there is human-like H&D stains in the brain tissue that shows you this. This is how the rabies-specific eosinophilic nucleons look like in the cytoplasmic neuron that vary in the brain regions and may not be found in about 30% of the cases. Although these lesions, they are considered to be specific and pathognomonic that clearly indicate the causative agent, but about 30% of the cases, maybe we won't be able to detect the Negri bodies by histopathological investigations. The clinical findings, as well as the long incubation period time, varies from 2 weeks to 16 weeks and even more longer. So an exposure to bite of the dog or to the bite or to exposure to the bat, if it is infected with the rabies virus, can take a long time to initiate the initial clinical signs and symptoms. In the prodrome of the non-specific symptoms, like fever, anorexia, you can feel some changes in the sensation at the site of the bite. You may not feel some sensation over there. So this is the early signs and symptoms. But in the later stage, once the viruses started traveling in and retrograde gone to the brain and cause encephalitis, you may feel some confusion. There's lethargy, and there is increased salivation due to the spasm of the muscle of the buccal cavity and tongue and all that. The most notable pain spasm of the throat muscles. On swelling, it results into hydrophobia. The patient or the affected individual become phobic to water because it does not have the ability due to the severe spasm of the throat muscles. It cannot swallow anything. It cannot even drink water. So it becomes photophobic. It becomes hydrophobic. And now the disease progresses to seizures because of the involvement of the brain. Then you see the individual may become paralyzed and ultimately go to coma. Death is almost invariably ensues. But with the rent of the life support system for individuals who have been exposed to this, survival is possible. So if you, in time, report to the clinician and the clinician started the immediate treatment or the clinical procedures, you can survive. Otherwise, if you are delayed from your life. So the laboratory diagnosis is very important. It's very interesting along with that of the treatment of the virus. They must the examination of the brain tissues using either the fluorescent antibodies to the rabies virus or histological staining of the negative body. This is very important. And then we can isolate from animal brain by growth in the cell culture. As with all other cell viruses, the viruses are always grown under the cell culture. They need live living cells because they cannot make their own macromolecules. They cannot make their own energy. They have to depend on the whole cell living energy. So they need the living cells to grow. So if you really want to isolate the cells, we can culture these cells, the virus onto the living tissues. In humans, we can also identify by the fluorescent antibody staining of the biopsy specimen. We can also isolate the viruses from the saliva, the spinal cord, and the brain tissues. It takes a long time. Requires a lot of sterilization procedures which are needed as a prerequisite for the isolation of the virus. And you can also identify the virus by the antibody to the virus that appears in the very later stage. The negative body is demonstrated in the corneal scraping, and then the autopsy specimen of the brain has also been reported. Now, treatment, you see, there is no antiviral drug available. Only we have to depend on the suppurative treatment. The supportive treatment is needed because the antiviral treatment is different than the treatment of the antibacterial therapy. Or antibacterial drugs, none of them can be used for the treatment of the viruses because the virus cells, they live within the cell of the human body. They live in the cell of the living cells of the host. So any drug that is being attempted to enter into the virus, to kill the virus, the drug also kills the host cells. So if you have a virus-specific drug, that makes sense. But if you don't have the virus-specific cells and the drug has the extra on the component of the host cells, it can inhibit the sense of the DNA, it can inhibit the sense of the proteins, then it can really cause damage to the host cells also. That's why we have to be very, very selective, and we are bound to the selective antiviral drug in case of treatment of viral infections. Now, what is prevention? This is most important. Now, in case of rabies, the prevention is more important than the treatment of the disease. Or there are two approaches. One is pre-exposure and the other one is the post-exposure. We can prevent the disease before the disease is exposed to you, or you are exposed to the disease. And we can also try to prevent the infection or spread of the disease in the human body post-exposure. Now, what is pre-exposure immunization is important. The high-risk individual group, they must be pre-immunized. There are 3 doses to be given at day 0, day 7, and day 21, or we can also go for 28 days. The booster doses are given at needed to the maintain an antibody rate of 1 and 5. The only vaccine, which is routinely used post-exposure, is the long incubation of all virus in vaccines, sufficient time to induce potentially. Now, what we have to do for the treatment, what we can do, we'll have to do two things together. We will have to give the hyperimmune serum, and we will also, this hyperimmune serum will not kill the virus, but it will try to neutralize the circulating toxin of the virus in the blood. And we will have to avoid this kind of infection with the help of the antimicrobial therapy. And we will also provide, some muscle relaxation for this organism, for this patient. The post-exposure immunization, it also involves the use of vaccines and the human rabies immunoglobulin, IgRIG. This is hyperimmune serum. The serum is having a lot of antibodies. And the vaccine will provide protection for the rest of the time, but for the immediate period of time to get the antibodies to be neutralized, we need the hyperimmune serum. This is what is needed. An example of passive, this is the passive and active immunization. Tetanus immunization should also be considered because if there has been trauma, there is a possibility of entrance of the spores of Clostridium tetani in the body. They are spores may germinate because of the presence of the wound, and they can be having synergistic effect on the body. So the immunization should also be considered. Almost, you see, the rabies virus along with that of the tetanus immunization is considered to be a best combination. That is all about. Thank you very much. Hello, and I'm sure that you all are well versed with the generalized structure of virus.